Hello, my name is Hannah and I'm a first year PhD student looking at adaptation in South African mole rats. When I say mole rats, most people will probably think of the naked mole rat or King Possible's Rufus, which is another naked mole rat, but actually most of them look more like this. And these are the haired mole rats or pes moles, and there are more than 20 species of them compared to the one species of naked mole rat. But the naked mole rat is a very biomedically interesting species, which is why it's far more well known. So for some context, they belong to the rodent family, and in the middle here we have rats and mice, and at the top in a genetically distinct clade we have guinea pigs and chinchillas. This is where we'll find mole rats. So despite being called mole rats, they are neither moles nor rats. They're more like very toothy guinea pigs. All African mole rats are found in sub-Saharan Africa and live in underground burrow systems. They range from being completely solitary to highly social, and again the naked mole rat is famous here for being one of the only eusocial mammals. Here we have the distribution of all African mole rats, and at the bottom in light blue we can see the Cryptomys genus. This contains the species that I'm interested in. There are 5 species: Cryptomys hottentotus, Praetoriae, Natalensis, Nimrodi and Mahali. And they have a wide distribution across all of South Africa, and are found exclusively in this country, which is quite unfortunate during the pandemic, but there we are. So how well a species is adapted to its environment is defined by the fitness of individuals, which contributes to the overall fitness of a population. Adaptation is a constant process, and thus species are not static, allowing for populations to adapt and persist as its surrounding abiotic and biotic factors change. Essentially, this project is looking at how these species are adapted to their environment, which is very unique. How are they adapted to the climatic conditions around them, and also the microenvironment of their enclosed burrow system. I'm aiming to look at genetic and metabolic traces and see whether there are differences between and within species as well. Unfortunately, the macroenvironmental side of things has been put on hold until I can get out to South Africa. But I thought I'd just very briefly show some preliminary maps that I've been making recently. So here we have distribution of known colonies, color-coded by the different species. And then I've overlaid the IUCN distributions on top as well. Now this is the same map with the color-coded colonies, but with different bioclimatic variables overlaid. So first of all, we can see that the biome type between species is already very varied, and even within species they're living in different biomes, so they're already experiencing different soil types, grass types and different aridity levels. And again, with soil surface runoff and relief, we can see the same patterns both within and between species. They're experiencing quite different conditions. Now, as I've mentioned, thanks to the pandemic, I've not been able to get out to South Africa and socially distance with mole rats myself. So I'm just going to leave that quite abruptly there and move on to what I've been looking at for the past year, which is specifically adaptation to the subterranean niche. I focused on whether there's a metabolic trace associated with living underground in a burrow and looking across the whole mammalian kingdom instead of just within the rodent family or one group in the rodent family. I also wanted to look at whether this is affected by how much time is spent in a burrow. So for example, as my pet rabbit is nicely demonstrating here, some species will use burrow tunnel systems, but as we know with rabbits, they'll also spend a lot of time above ground and will forage above ground. This again compares to the mole rats, which will spend their entire lives in their burrow system and will never see the surface or daylight really. So I'm interested to see if there'll be a difference between these two lifestyles. So burrows create a localized contained environment around an individual or a group of individuals, and will have certain conditions such as high humidity, low oxygen and high CO2 concentration, low light levels, and small fluctuations in temperature. This will require specific adaptations in order to be able to live in such conditions particularly if they're spending a significant amount of time underground. These adaptations have been found in individuals and individual species groups. For example, hypoxia tolerance to low oxygen levels, has been found in a different mole rat group called the blind mole rats. And this is a different hemoglobin shape which has a higher affinity for oxygen. A slightly different hemoglobin shape has been found in the mandarin vole, which again has a higher affinity for oxygen. And voles will spend time above ground and below ground. They'll spend most of their waking time above ground foraging. So we can see that this intermediate group are showing some adaptations already. Low BMR has been found in subterranean rodents such as the African mole rats, and again the blind mole rats, as well as the pocket gophers, which are a subterranean rodent family from North America. This is thought to reduce gas exchange, so there's less CO2 being produced and oxygen is being used up much slower. There'll also be a lot of morphological adaptations to living in a burrow. There's obviously a size limit and also the need for a particular shape in order to move effectively through the burrows. There may also be the need for digging appendages if that species is creating the burrow system itself. In order to get data, I started with an existing database from this 2017 paper, which had the BMR and body mass of over 800 mammal species. I then created definitions for my different lifestyles, which you can see here. We have terrestrial species, which will spend their whole life above ground. It's fairly easy to define. Fossorial species will use a burrow at least once in their daily cycle, but will be spending a significant amount of time in the burrow as well as above ground. And finally, subterranean species, which is where the mole rats would be included, will spend approximately their whole life underground and will pretty much always be digging their own burrow system themselves. I then added the 20 plus life history traits, which was the very time consuming bit. And then for the analysis, which will be the next steps, I'll be looking for collinearity between these life history traits and then finally running a PMM analysis on the full data set. So I do have some preliminary results. This is the PMM, the results for a PMM that was run on roughly about 400 species. I've been working on this database with someone else in my lab group, so we've split the database. And so I've only run this on my half. But we can see that phylogenetic heritability is very high, which shows that BMR is strongly determined by phylogeny. And this is further backed up by this table, which shows the results of the PMM. We can see that the model with the higher ranking is the one where phylogeny is accounted for, and this is a much better fit compared to the one where phylogeny is not accounted for. The top ranking model suggests that body mass influences BMR most strongly. This is really well known. Body mass has a huge influence on metabolic rate, so it's really no surprise that that's the top model. But the second highest includes terrestriality as a factor, so it's potentially having some influence. And this is further shown by these graphs that I've made, where we can see that subterranean fossorial species have a much lower BMR than terrestrial species. And there's actually not much difference between fossorial and subterranean, interestingly. We can also see that subterranean species have much less variance, and this is potentially because there are fewer of them, but it could also be because of convergent evolution. The constraints on living underground are quite strong, so that might be influencing this. As I said, this is only part of the data set, so it'll be interesting to see how this changes when there's more data added and whether these differences are significant, and how the PMM might change when the full data set is used as well. So there's a lot more to do, obviously. I'm only in my first year and the pandemic has been going on for the whole of that year, which I'm sure is the case for a lot of people. It's rather slowed things up, but it means there's a lot more exciting things that are yet to come. And with this project, I hope to add to the data exploring adaptations of species in a variety of terrestrial climatic conditions, as well as further understand how species are adapted to the subterranean niche. Current knowledge of physiological traits is representative of only a limited range of climates in which terrestrial animals will be required to operate. So with this project, I hope to add to this body of knowledge and help understand how these species are adapted to their unique environments, which may then potentially help advise conservation policies should these species ever become endangered. I'd like to thank my supervisors and my collaborators in South Africa as well as Jack in my lab group who's been helping me with the database. And thank you for listening.